Hi everybody. We are ready to pick up where we left off in lecture. We had been discussing, I think, the last time we met in class about the skeletal muscle and specifically talking about some of the components of the sarcomere, which you remember is the structural and functional unit of the myofibril, of which there are many of these within each skeletal muscle fiber or cell. You might recall we have the thick and thin filaments. The thin filaments are basically anchored to the Z lines, which make up the periphery of the sarcomere. And pieces of that actin, remember, extend toward the center of each sarcomere, with the myosin, the thick myosin filaments shown here in pink, in the very center. We also defined some structures within the sarcomere, like the I band, which you remember lie on either side of the Z line. And here we have just the thin actin filaments, while the A band is showing a darker striation, part of which refers to an overlap of the two. So here we see the actin and the myosin overlapping here and here. And that refers to the darker, I can't go back. Yeah, it shows here the darker purple aspects of the sarcomere. That's where the two overlap. And in the very center of the A band is where we have just the myosin. Remember, the myosin is anchored here in the very center at what's called the M line. There's also one over here, even though it's not shown. So we had talked about that at the last time we had gotten together prior to the COVID issue, right? So go back and review that anatomy of the sarcomere. Really important to know those different parts, myosin and actin, how they're oriented with regard to one another, and then the I band and the A band and the Z line and so forth. This is a magnified electron photograph, probably from a compound light microscope, of a sarcomere. So hopefully you recognize these darker blue structures. Do you know what those are? Those are the Z lines, right? There they are. So between the Z lines is the sarcomere. We have just the lighter components. Remember, on either side of the Z lines was the I band. That was just actin. And here's the A band in the middle, the darker component. Part of this, again, is showing where the two overlap. And in the very center, we have just myosin. Here we're looking at the last couple slides. Again, I'm not going to say a whole lot about that. We've talked about this. So you should be able to now see sort of the structures on the diagram, right? And how that corresponds to the reality within the actual section of stained skeletal muscle within that sarcomere. What we're going to do next is we're going to zoom in on where the myosin here and the actin come in real close association with one another, because they're going to start to interact as the skeletal muscle fiber and the myofibrils within it begin. So here's our myosin. This is that purple structure here shown in the oval, right? There's another one here, another one here. It's more on the sides. And then here's the thin actin in blue. Looks like there's about 8 of them here, right, in this diagram. We're looking at 4 of those here. So here's 1, here's 2, here's 3, here's 4. And we note that the myosin, here's that thicker filament that has these so-called head parts of the molecule, this protein, that stick out in all directions. Some above, some in front, some below, some on the other side that we can't see. And then we've got the actin, of course, above and below the myosin. We also would have one here in the front that we can't see, and there'd be one tucked in the back here too, because this is 3-dimensional, remember. And you'll notice again that in addition to the heads here, like paddles that stick out from the myosin, the actin filaments, the thin filaments, are made of this sort of double strand of globular proteins here that are wrapped in sort of a helix, right? Well, that's kind of interesting. If we look on the surface of this helical actin, we see this darker blue colored structure, sort of like a strand. And then every once in a while you see this 3 subunit structure here. They're scattered around. Okay, so the rod-shaped blue strand here is referred to as tropomyosin, and occasionally we run across this 3 subunit protein called troponin. And together these two structures make up what's referred to as the troponin-tropomyosin complex. And I can see from the video here that I've kind of cut off the last part of this sentence, but if you go back in some of the non-narrated PowerPoint slides, of course, anything that happens to be cut off by the video you can access. All right, so we need to understand again the different parts of the myosin and the actin so that we can begin to understand how they interact with. Let's make special note of this diagram that shows how the tropomyosin molecule here, the blue strand, is covering that helical actin, and it's blocking what's referred to as the myosin binding sites. So every once in a while you see a little pink structure here on each of these circular structures, ovals, and this is where the myosin paddle here, the head of the molecule, is going to try to come up and grab onto the actin if it can, but now it can't because the tropomyosin is blocking those binding sites, right? The paddle here of the myosin can't come up and grab on to those actin proteins as we see it on the diagram. Therefore, that particular sarcomere can't contract.